Okay. Any residual? So Brian Yandell again. I'm going to do a demo of some of the packages. It's sort of an overview of more of the strategy than details of the packages. You guys have been going for a couple days now, so I'm not going to bore you with it. I already bored you with some of the details. I want to do a little lighter touch here. And again, we're using R. That's a common theme in this course. And as you've no doubt heard at the beginning, R is a nice language to take concepts and develop them into usable algorithms in a fairly ready way. So I first want to, let me just run this, okay. So I gave you this, this is on the web page for the course. And if you are, and I'm going to be using a system called RStudio, which is, I only started using it very recently actually, but I like it a lot. It's allows you to sort of have everything in one place, as you'll see in a minute. If you have questions about QTL studies, then I would point you to this RQTL site, as I did earlier. And in order to install packages, there's basically a routine in R called install packages, and you give the name of the routine, and it'll bring it in. There are a couple other routines that that we're using. So the hotspot and the causal pairs that Elias will be talking about a little bit after the break, to the next break, are both in QTL hot. And then there's another package for the network study called QTL net, which draws on some other routines from bioconductors. So there's some extra steps here to get that in place. And then the yeast data from Bremen-Krugliak is available in, so it's publicly available. You can find it. The version we have here was cleaned up by Bin Zhang and Jun Zhu and Eric Schatt when they were all at Rosetta. And then Elias and I organized it into a library, so it's available on GitHub. So there's some extra steps to get it in here, but if you just follow these things, you'll be able to install it on your system. There's a little extra if you're on a Windows machine, a little extra steps you have to do. Okay. So so I'm going to go over this example here, and let me just run the example here. So I'm using this system, this RStudio, which has the session. The R session is down here, and the commands you run are up here, and then you can get plots and things over here. I'll show you that in just a minute. And there's a, what I'm going to do is run through these chunks. Let's just run the first chunk. Okay, oops, yeah. Okay, so the first thing I'm going to do, this suppressed message is getting rid of extra stuff on the screen, but I load up the QTL library, I load up the yeast library, and then I'm just looking at a summary of the yeast data set. So it's got 6,000 phenotypes roughly on 16 chromosomes, and there are about 3,000 markers involved in this study, 112 individuals. Okay. Now then we go to the next chunk. And this is the genetic map. And then if we go to the next chunk, I've got, okay, so what you can find in the, where is it? Lost it here, okay. What you can find on the web page for the course is this document. I don't remember if it's a PDF or, I think I put it as a PDF. But I just regenerated it here, right here. And it goes through these steps, and you'll see code interspersed with the figures. And basically, you can cut and paste out of here to go through the examples. So here's the genetic map. And you'll see that there's markers spread around. Some chromosomes have dense markers, some have gaps in them. So that gives you an idea about what we're working with from the genetic information. And because we did some little work ahead of time, we're going to look at a particular open reading frame on chromosome 15, which corresponds to a particular gene. What is the gene? The gene is named PH7, and the name of the open reading frame is YOL0 whatever 84W. Okay, and it's located on chromosome 15, and I'll show you more detail about that as we go along here. So I showed you where the markers are located. Now we're going to look at the genotypes, but just on chromosome 15. So we've taken the individuals in chromosome 15. We've sorted them based on their this particular open reading frame, which is called here TF.ORF. So this is the open reading frame that we're focused on, and then it's actually located right near this marker right here. And so you see that this is like a back cross, basically. So one genotype is red, one's blue, that the reds tend to be on the bottom here, the blues tend to be on the top. There's very good separation here based on the phenotype of the genotype. So, and again, as I was talking about before, these gaps here where you go from red to blue, that indicates a crossover event. There was some sort of recombination that happened. And sometimes there's double recombinations involved here. Okay, any questions so far on?
Brian, the white blips are missing genotypes?
Yeah, the white blips are missing genotypes. So let me go back over here and plotted the map. I'm just going to go through these commands that I just showed you on this other picture. Let's go to the next. Okay. So there's I just recreated it right here, this genotype image. Okay.
So you sorted on the expression of the transcript of that gene?
That's right. Yeah. Okay. And so you might expect that since there's a really good match here that we might actually see when we look at the map of this gene that it would come up on chromosome 15. So let's verify that. Okay. And here it does. So here I've done the map. So how do we do that? So this is first thing I do right here, this line right here, is I calculate genotype probabilities. I basically fill in for missing data. This is a part of our QTL. And then I do a scan, a one-dimensional scan. And as I didn't mention, as I was coming out in the discussion in the hallway, we're doing a one-dimensional scan here saying that there's just a single QTL. And yes, there are undoubtedly many QTLs that affect most traits. You know, we're working on mice that were sacrificed at 10 weeks, and undoubtedly there are thousands of genes that affect any particular trait. But a single QTL model actually does pretty well to a certain degree with caveats. So, and we see here in this case that basically we're seeing one signal and the rest looks like noise pretty much. And then I'm just doing the same thing again, but focusing on chromosome 15. So I did a plot here, and then now I'm doing a plot just looking at the chromosome, and I identified that chromosome earlier. I guess it's probably up here. Yeah, it's identified here as chromosome 15. Okay, and then I brought in a threshold based on permutation tests. So there were permutation tests that were done. I'm not showing you those, but they are in the, there's a vignette for this package, and you can go to that vignette and see the permutation. You basically do the scan one and ask it for to do as many permutations as you want to do. But in the interest of time, I'm not going to show that here. but it's a permutation threshold of 5. And then this is where the gene is located, and so it looks to be cis-acting. It looks to be locally acting. Looks pretty good. Okay. All right. So now we're going to look at hotspots. Okay. So now we're using the, so up till now it's just using the QTL package to do genome scans, and we just picked one in particular that we were interested in. Now we're looking at the hotspots, and so now over the whole genome, I'm just focusing on a couple of chromosomes here, the ones that showed strong hits in my previous talk. So here's chromosome 14 with a lot of hits here. A little different threshold than was there before, so that's why the numbers are different. And then this corresponds to the that extreme threshold of nothing over the whole genome. So this is, again, at about 82, I think, or 96, I guess was what was used here. Okay. Now, the next piece here is going to just focus in on chromosome 15. So now, again, we're looking at hotspots. Let me just say a couple things here. So there's some things I'm not showing you here, again, in the interest of time. But basically, what was done between looking at a single trait and looking, in this step, is there was genome scan across all 6,000 traits. And then for each of the traits on each chromosome, just the peak area was preserved. So the peak and anything within 1.5 lods of the peak was preserved, and everything else was thrown away. And that's all preserved in this object called highlod.orf, which is actually saved in the package. It cuts down the size of the object by a factor of at least 100, maybe 1,000, because a lot of the, and so we're only looking at peaks that are above the Lod threshold of roughly 5 here, and only areas around that peak which are within 1.5 of that high threshold. So this is this high Lod object, and so we can actually get a manageable size object from that. That's one of the things you have to deal with, is you can't just keep 1,000 scans or 6,000 scans or 40,000 scans lying around. They take up a lot of room. Okay. And then we get the hotspot size from that object using a particular threshold, and then we get a summary of that. So here's a summary right down here showing what the maximum size were for the different hotspots, and I showed you the picture. I can go back to it here. So here you got chromosome 14 at 259, chromosome 15 at 125, and so on. Okay. And so again, one of the nice things about R is that you can create these objects. You can look at summaries. You can do plots in a fairly straightforward manner, just adding a few little annotations to make it fit your own needs. Okay, let me go back to the plot we have here. Okay, so then here's again chromosome 15. We got a hotspot here, and now we want to look at that hotspot a little bit more. Okay, so the next thing we're going to do is look at causal pairs. So this is getting into, this is anticipating the next talk, and connects a bit with what Mark was talking about last time. So we're going to look at, take this particular open reading frame and find the candidates for that, for its regulation. So let's see, and then find which of those co-map. So let's see, what is, it probably says it over here. Okay, so that's a, Okay, so this gives me information about this particular gene. It's it says gene here, but this is actually open reading frame. It's physically located in chromosome 15. There's its physical position, and the peak is here, and there's the peak position, and there's the LOD for the peak. Okay, and then I also pulled out the things that co-map to it. And there are basically 407 different traits that map also to chromosome 15 that have peaks somewhere within a few centimorgans of this peak here. Okay. So now let's see. The next thing we do is we're going to now do a bunch of tests on all those 407 traits. So we've got a this particular open reading frame is located on chromosome 15. We've got 400 other things that co-map to the same, basically the same position or close to the same position within shouting range. And we're going to, for each of those 407, we're going to do a causal test that Elias is going to explain. All right, so we do all of those. And see what's the size of this target peak. I'm going to see if it's worth. Okay, it's 113. Okay, so. Okay, and so after doing some magic here, we're basically coming up with causal calls for each of these traits. And now we're going to focus in the next batch here on the significant ones, because I'm just pulling out the first couple ones. This is not significant. It's causal. It's indicating it's causal call, but it's not significant. None of these. Maybe this one looks interesting. It's got a p-value of 0.009. But let's look at the next set here. Okay. So let me just review here. So I've done causal model tests, and then the rest of this is just some bookkeeping to pull out what I wanted, which is basically the p-value and the call as a word. And now in this next piece that I just ran, I'm just going through and sorting these by the p-value, and then indicating which ones are significant at the 0.001 level, which ones will be significant at a slightly more liberal level, and then I'm giving the name of the gene as well. Okay. So that's all I'm doing in this next set. It's mostly bookkeeping. Okay. So now I'm going to take these ones at the very top here, and I'm going to run scans of these traits. So here's the alleged causal trait for the gene PHM7, and then here's the other half a dozen, which also co-map to the same position, roughly the same position. Okay, so I just want to look at what's going on with these.
Brian,
Any question? Yeah.
Do you demand that they not be cis?
No.
So you allow for a cis co-mapping trait in the causality call?
Yeah, and these don't even have to be expression traits. They could be anything.
Okay.
Okay. And so these are the same lines. I'm just focusing in, not on the whole chromosome, but the first 100 centimorgans of the chromosome. Okay. So that's, so we have these causal calls here. That's the main result from the causal. And what we did with Mark was we actually went across every possible candidate. Now here I'm just looking at one candidate, this PHM7, but we did every possible candidate and did these same type of tests for each of those individuals, and to come up with that in the story that we talked about yesterday. Okay. Now I'm going to shift gears from hotspots and causal pairs to looking at causal network. So now I've identified half a dozen traits that are of particular interest to me, and what I'd like to do is now form a causal network with these. Okay, let's see. And do I actually want to run this? I think what I want to do is I'll show what it is. I'm not going to actually run it. It'll take a few minutes to run it. But what I did here in the code, and it's also in the piece that you can look at here. So right down here, this piece right here, I'm not actually running this because it takes 5 minutes to run it for this data set. It takes a lot longer for the mouse data. But I ran that elsewhere and I saved the results. And so I'm not just going to load up the results. So I could do it by cut and paste from here. And then it'll, in a moment, produce a graph. Okay, there's a graph. And I'm going to do a, oops. Okay, so what I used is yet another graphing package called iGraph, which is part of the R distribution, which is why I used it. But again, you've seen now several different of these graphic packages, and there's a certain amount of convergence of what these things do. Now let's see, why is this not working? Where's the plot? Okay, I'm not getting a plot. Okay. Anyway, let me show you this one then. Let me see if I can make this a little bigger. I don't know if I can make this the whole thing. Nope. Okay. So it's a little small here. I should be able to get a plot that I could actually manipulate, but it's not coming up. Anyway, here's in yellow is the driver, and as I hope you can see, maybe we can see it better. Oh, there it is. There it is. Okay, here we go. Just took a while to come up. Okay, so here's the in yellow, and there's arrows going out from it to all of these other pieces. Okay, I didn't set those up ahead. I just put all of these. I put these 6 traits into a causal network calculation. It uses the QTL stuff. Elias is going to talk about this a little bit. And it came out with these being all downstream, all 5 of these other ones in green. Here's the chromosome 15, excuse me, QTL. And then there's a couple other QTLs that came into the story. And then we have some other causal relationships that came out with this as well that could be, and this basically forms a test bed for further investigation. So we've got, this appears to be both have a direct and an indirect target, and this one as well as an indirect target, and this one is doubly indirect, both coming from this way and through this pack this way. There's a couple other QTLs that come up here. Yes, question?
I'm just curious, this causal network, is it built using structured tree model?
Structured tree models?
Structured equation model.
It's related to structural equation models. At least we'll get to that. Yeah, yeah. So it's, yes?
So, I'm sorry, so the green and the red, does that represent that those genes are being repressed, upregulated and downregulated, or are they activators and repressors themselves, or what does that represent?
So there's no direction.
Okay.
So, I mean, we could put direction here. It's just not built into it. But, this was the driver. So this is the PHM7. This is the ORF for PHM7. And these others came up as being of the 407 that were mapped to the same place, these 5 came up as being most significantly downstream from this yellow one. So there's these 5 in green. And the red are the QTLs that came up in the analysis. So those QTLs were not specified in the analysis. What was specified is the name of these 6 traits. And then the network emerged from the magic of running it. Yes?
So maybe Elias will get more into this later, but I was just wondering in terms of the output of the R package, if a green to green, if the approach that's looking for the best network cannot determine whether a green to green edge should go in this direction versus that direction, like if those are both equally good solutions, does it just output an arbitrary one, or will it show both directions?
This is actually what's plotted here is the result of is the network, but it's actually a Bayesian model average over the edges.
Okay.
So pretty much if you have, for example, a mutual top test.
Yeah.
Okay. But if you have something that have about the same probability of going one direction and the other, when you get to the Bayesian model average one, you are going to pretty much rule those out because you can actually select the threshold.
Oh, so you won't see any edge in that case if it doesn't know, if it's about 50%
Yeah,
chance.
usually we set the threshold for the Bayesian model average one higher than 50%. So pretty much situations where it's, they go both ways, it doesn't show up. But I'm going to explain that more in detail.
Yeah.
Thank you.
And if it's not clear, what's clear.
Yeah, so you get no edge if there's no relationship between them or there's equal in both directions. Yeah. And there's also no cycles. We don't allow cycles in this. Okay, so that gives you an overview of the packages. Elias will get into some more detail about some of the causal approaches and also some of the algorithms behind the causal stuff here. Okay.
Are you done?
Yeah, I'm done.
Thank you. I think we all like this.
Okay. I'm sorry I got messed up on that one spot. I did figure out what I did wrong. This is permutation. When it says here above a 1 trait threshold, this is the threshold right here determined from from looking at single traits. So this is supposed to be a single trait threshold, but I made it short and I confused myself. So anyway, all right.